Hello everyone, welcome back to my channel Microbiology Easy Notes. If you like this channel, please like, subscribe, comment and share. Today's topic, Vibrio cholerae. In this video, we will cover the morphology, classification, pathogenesis and lab diagnosis of Vibrio cholerae. Vibrios are among the most common bacteria in surface waters worldwide. It is responsible for cholera disease in humans, which is an acute diarrheal disease. Morphology of Vibrio cholerae. It is comma-shaped, curved aerobic rods. Size is 2 to 4 micrometer long. Usually it occurs singly, but sometimes in pairs also. For motility, it has single-sheath polar flagellum. Some strains possess capsule and are virulent in nature, while some do not have capsule and are non-virulent. Gram staining. Vibrio cholerae is gram-negative. On prolonged cultivation, vibrios may become straight rods that resemble the gram-negative enteric bacteria. Sporulation is absent because they do not form spores. Apart from flagella, villi and fimbriae are also present, which helps in attachment to the intestinal mucosa. They can be anaerobe or facultative anaerobe. Vibrio cholerae can survive in salt-rich environment, but they cannot survive in acidic environment. Classification of Vibrio cholerae. Vibrio cholerae is divided into more than 200 serogroups on the basis of structure of the O antigen of lipopolysaccharide. Among them, a subset of strains belonging to serogroups O1 and O139 can cause cholera and epidemics due to their ability to produce cholera toxin. Serogroups that are not O1 and O139 collectively referred to as non-O1, non-139 and they lack cholera toxin. Non-O1 and non-139 responsible for small gastroenteritis outbreaks. Based on biotypes, O1 strains can be further divided into classical and Altor. Altor strains are more efficient at host-to-host transmission, survive better in the environment and the human gut. Altor also have a higher occurrence of asymptomatic than symptomatic carriers compared to the classical strains. Furthermore, O1 strains are divided into 3 serotypes, Ogawa, Inaba and Hikujima. While the Ogawa and Inaba serotypes can co-circulate during epidemics and are capable of interconverting, the Hikujima serotype's occurrence is rare. Pathogenesis of Vibrio cholera. Cholera is transmitted by the fecal-oral route. Vibrios are sensitive to acid and mostly die in the stomach. Surviving virulent vibrio adhere to and colonize to the small intestine. Here they secrete the potent cholera enterotoxin which causes diarrhea. What happens when Vibrio cholera binds to the epithelial cells of intestine? Once Vibrio cholerae reaches to intestinal epithelial cell, it releases AB5 type of cholera toxin. AB5 binds to GM1 receptor present on epithelial cell with the help of B subunit. Receptor allows entry of AB5 toxin through the process of endocytosis. A and B subunits get separated in the endoplasmic reticulum. Further, A subunit separates into A1 and A2 subunit. A1 subunits bind to G stimulatory protein and activate adenylate cyclase. Due to this activation, CAMP releases in excessive amount. Excessive CAMP binds to CFTR and phosphorylate it. CFTR is a specific channel which helps in chloride ions entry and exit. But after phosphorylation, CFTR expel out chloride ions from intestinal lumen. To create osmolar balance, water also come out, which shows in the form of diarrhea. Lab diagnosis of Vibrio cholera. The diagnosis is suggested by severe watery diarrhea, which looks like rice water and smell fishy. For rapid diagnosis, a weight mount of liquid stool is examined microscopically. Stool or rectal swab samples is cultured on selective media like TCBS agar and other selective and non-selective media. On TCBS agar, Vibrio cholerae produce yellow colonies in the green background. There is a slide agglutination test also which can confirm the Vibrio cholerae presence in the sample. In this test, bacterial culture in saline water is mixed with specific antiserum. If clumping occurs within 30 seconds to 1 minute, it indicates positive reaction. Biochemical tests are also there to confirm Vibrio cholerae. It is oxidase positive, catalase positive, and string test positive. What is string test? When a colony of Vibrio cholerae is mixed with a drop of 0.5% sodium deoxycholate, which is a bile salt solution, it will form a viscous mucoid string. For serological diagnosis, fluorescent antibody test and retrospective serological diagnosis test are available. For molecular diagnosis, the polymerase chain reaction and additional genetically based rapid techniques are present.